Is George Dizikes. George is the director of the Knoxville Regional Laboratory for the Tennessee Department of Health, and he's been there since 2015. Prior to that, he was section chief for the Newborn Screening Laboratory for the Illinois Department of Public Health from 2011 to 2015. And his presentation title is First Year Experience Performing Newborn Screening for 5 Lysosomal Storage Disorders in Tennessee. Take it away, George.
Okay, good morning, everyone. Thanks for being here, and thank you for the organizers for asking me to come. I'm going to present a little bit of data from Tennessee on our testing for lysosomal storage disorders during our first year. This is a picture of the laboratory, which is in Nashville, and it houses both the laboratory and also the follow-up program, which makes it very convenient. All right, so in 2017, or actually in 2015, the Genetics Advisory Committee voted to add 5 lysosomal disorders, and shortly thereafter, after XALD, which is not a lysosomal disorder, but after it was added to the RUS, they also voted to add that. So development of those tests began, and July 1st of 2017, we began screening for those 5 disorders, and then in April 30th of this year, we started screening for XALD. I'm going to just say something briefly about that because I think a lot of newborn programs are struggling a bit in getting ALD screening up and going. We decided not to try to multiplex that with LSDs, or LDs as they're called now, because not all of them are storage disorders. Instead, we use a separate punch, and it's a very straightforward assay. We get internal standard as an analyte-specific reagent from PerkinElmer for C26. That's added to the methanol that's used to reconstitute a separate punch. It incubates for 30 minutes, and then without drying or any other processing, it's just injected directly into an LC mass spec, goes through an HPLC column, and is run under negative ion mode. It takes about 1 minute per injection or 90 minutes per plate. And the only issue with this is the cone gets pretty dirty. It's almost like you're spraying cooking oil onto the thing, which you essentially are. So we have additional cones that are ready to go so they can be swapped out when the signal starts degrading. We use the same tandem mass specs. They're TQDs that we use for LSDs, and they can be run, of course, in separate runs, but with just changing the software settings. During the validation, we found 1 positive, and since then we haven't found any additional ones. So before beginning testing for the lysosomal disorders, I just want to show some data on consideration here. It's, as we all appreciate the fact this is an enzyme assay, and it requires a certain period of time during the incubation to generate enough product to be able to detect it with the mass spec. So these are data that I generated when I was at the Illinois Department of Public Health, and it shows that for Pompeii, or GAA, incubations of 3 or 17 hours generate essentially equivalent data. And why this is important is that in the workflow of the day, if you can run the incubation during the day and process it and get it on the mass spec that night, you can get data the next day. If you go more than 3 hours, you're going to essentially commit yourself to an overnight incubation, which adds an extra day to the assay, and if you're going to then repeat that, it adds 2 days. So this shows the equivalency of a 3 or 17-hour incubation for GAA, which is Pompe, IDUA, which is MPS1, and at the bottom, GALC, which is CRAB-A. And as you can see there, you don't get nearly the amount of product generated during an incubation, and that you actually have to go out to 17 hours to be able to separate the low normals from abnormal specimens. So if your lab is only going to do Pompe and MPS1, you could do 3-hour incubations, but if you're going to do CRAB-A as well, then you're pretty much committed to a 17. Now you could try to split the workflow, but that causes additional problems in keeping track of where you are with regard to initial runs or repeat runs during the day. And we decided to just go with 17 hours for everything. The other thing is that CRAB-A is the most time sensitive of these, so the others, an additional day or two of incubation time in processing isn't going to make that much of a difference. So after doing the validation, we decided on the following cutoff levels for the different enzymes. And what we do is we have an initial cutoff, and these are based on a % of the daily median. Now, some people will use mean. We use median because it tends to minimize the effect of the outliers. So, for example, in Pompe, if something comes up at less than 20% of the daily median value for the enzyme activity, that gets repeated in duplicate, and then the second cutoff would be at less than 15. Something between 15 and 20 on the repeat would be borderline. So you'd repeat the borderline, or you'd ask for an additional specimen for the borderline. That comes back, and if you again get borderline, you consider it a positive. Otherwise, it's what it is. As you see, for CRAB-A, there was no borderline, and that's to minimize the amount of time that's being spent on doing the initial evaluation. Now, about halfway through this process, we reevaluated the cutoffs and are now using what's shown on the bottom table. So here, as you see, we don't have a second level of cutoff. There's an initial cutoff, and then there's a repeat done in duplicate with the same cutoff, and then if it again scores below those, it is called out, or it's referred for additional testing. Now, this is the algorithm of our testing. It's, as you might expect, you run an assay. If it's positive, you go over to the left. If it's negative, you report it as within normal limits. If it's a second test, then it goes down the left lane here, where if it's a repeat as abnormal, it's sent out to our family health and wellness people, which are the follow-up. If it's a CRAB-A or a Pompe, then the only difference in following those up is the additional testing is initiated more expeditiously. Particularly for CRAB-A, we will send punches out to the reference labs, which are Mayo for second-tier biochemical or PerkinElmer for sequencing, and they'll hold those until we get the repeat results, and if those are still out of range, we tell them to go ahead and run those, and if they're within range, we tell them to return the specimen or destroy it. In the case of the other disorders, we'll wait until we get our repeats. So the strategy, at least for the first year here, was anything which was abnormal was referred out for second-tier tests and also for DNA sequencing. Now, this is kind of an expensive proposition. The sequencing can be up to $1,000 per specimen. Second-tier testing is in the like $100 or so. So as you can see here, you've got Pompe has second-tier testing done at Mayo, and that's the ratio of the creatine, creatinine, and GAA. CRAB-A is cytosine, Gaucher is glucocytosine. Fabry doesn't have a second tier yet, and MPS1 is the dermatan and heparan sulfate levels. These take several days to do, and sequencing is up to a week. And this generally doesn't cause a problem except if you're trying to evaluate a potential Crab-A for a transplant. So these are the results we got for the first year. And actually, the presumed positives, which would be those that fell under the cutoffs, are about 140. For Pompe's, we had 3 infantile, 8 late onsets. You can see the carriers and the pseudodeficiencies. MPS 1, we found 2. Crab-A, no infantile, 2 apparent late onset, and these are probably the toughest. Really? 5 minutes, okay. Those are the most difficult cases, I think, among the lysosomal disorders to evaluate, and those are the late onset in general and late onset Crab-A in particular. So one of the things we started doing during this is to use clear. There's a reference here to a very interesting paper out of Mayo and Kentucky on using clear for their first year of evaluating specimens. Holy crap. I thought that was off. Okay. So I would recommend whoops, what happened here? I would recommend anyone who's going to do this and wants to utilize clear, and I would recommend exploring the use of clear analysis to refer to this paper and to get in touch with the people at Mayo. Now, I'll go through this fast. What it just shows is what happens when you apply clear to evaluations that were otherwise done conventionally. So the upper part, these are the Pompe or GAA results ranked by enzyme level. If you then rank them by the clear scatter plot score, which is in the column over fourth from the right, you see it makes a little bit of difference, not a lot. What it basically does is the important ones, the infantiles are in yellow, the ones in blue are the later onsets, and it tends to bring them up higher so that if you wanted to evaluate just those, you could pretty well eliminate the pseudodeficiencies in the carriers. For MPS 1, it's not quite as clear. The 2 we had come up at the top with the lowest activity. The ones that are in blue are carriers. Those aren't necessarily what you're interested in, and your advisory committee and the people who deal with these are going to have to decide what you want to deal with. Do you want to call out carriers? Do you want to call out late onsets? These are questions which I think are not fully resolved. This shows when you do clear analysis versus just by enzyme, and it doesn't make much difference. Crab-A, again, the ones in yellow are the late onsets. They don't really stand out, and you get a lot of pseudodeficiencies and carriers that pop out based on the cutoff. If you drop the cutoff below where we currently have it, and that's pretty low compared to what other states are doing, you're going to miss potential cases. Fabry is its own problem because there's not an accepted second-tier biochemical test, and what we have, and you get a lot more Fabry's than you would expect, and they tend to cluster based both on enzyme and by clear analysis. Clear analysis, as you see, the normals tend to drop to the bottom, so I think it's well worth pursuing this. The problem we have, and people starting out will have, is that you're not going to have enough data in the clear database to do a very precise analysis. And then Gaucher, we didn't have any positives. Things are just kind of scattered throughout, and all of these are just carriers anyway. So ongoing, we're going to continue to screen for the 5-plex using a flow-injected mass spec. We're going to do clear analysis using our 5-plex, which are the 5 enzymes we're currently assaying, possibly a 6-plex or a 10. The 10 adds 4 LPCs to this. Second-tier biochemical tests, rather than doing them on everything at a cost of $100 or $200 each, you do it based on what CLEAR indicates. Same is true with DNA analysis. And then later, we're planning on adding SMA. So these are the folks that actually do the work. I'm just telling you about their work. Chris Dorley is the lab manager, and this is her staff. Amanda Ingram is in charge of follow-up. Yin-Mei Lee is our data person, and we're all in the Department of Health in different divisions, lab services and family health and wellness. And here's a picture of the lab staff, and thank you.
Probably have time.
Almost.
Questions? Time for a couple questions.
Your slide showing the sort of, could you go back to that slide showing the total? Okay.
Okay, which one? One of these?
Back to the summary slide of your findings. That's the one.
Okay.
So we've got 22 in the MPS 1 with variants of uncertain significance,
Right.
and 9 in the Krabbe category of uncertain significance or pseudodeficiencies. I know they're mixed. But what would the laboratory propose to do with the variants of uncertain significance for those two conditions? So you've got an enzyme deficiency, then you did molecular and it didn't clear up the story. Then what?
Well, this is the problem.
Exactly. That's why I'm asking the question. And it's not unique to Tennessee because it's in both the Missouri paper and the Illinois paper, but it's an extremely significant problem for me because the laboratory tends to pass that on to the follow-up team, the clinicians, and we don't have any revealed knowledge about this.
Well,
and so
I don't.
It needs to be discussed before states start doing this.
Right. I don't mean to disappoint you, but we don't know either.
And I didn't mean to point out that that was a deficiency in any way, but it's a problem
I really
in
think,
the field.
Okay, I think the current, now, there's two ways of looking at this. One, you're never going to know what those mean unless you bring them out, follow up people for a long time, and see what happens.
That sounds like research to me.
I think, well, personally, I think screening for Krabbe is research. Okay. And Dr. Laney Ross said Illinois would fully agree with that, and we've had talks about that. But, and the whole process by which a state can mandate research is for a different group or a different time to discuss. I think the best solution, and it's not a good solution, is to better define what it is you want to see. What are you looking for? Are you looking for like early onset Krabbe? If you are, then you can do clear analysis with cycosine as a second tier. And if the cycosine is elevated, your day is over. You don't do DNA analysis.
That's a news story, indeed.
And I think for some of these, DNA analysis doesn't clear things up, it just muddies the water.
Right.
And there should be less of it, not more of it. It's not a solution to some things. And that's all I can say about that, that you just avoid the problem.
So I just want to be on this soapbox. I put a bullseye on my chest just to say that what the state mandates is not research, and what we end up with is doing research. And what gets caught in the middle are the patients.
Well,
and
if
what
the
to
state,
do with those patients where we don't have an answer.
Legally, if the state mandates it, it's not research.
That's right.
Okay, but, you know, it kind of looks like research, and walks like research.
Okay.
Okay, all right,
Thank you, George.
Thank you.